Right, okay, second lecture now on immunometabolism. Let me fly through it, and I'm going to keep this tight because I've got something that's looming. A lot of detail in the first one, so we did cover lots of ground actually, so this next one shouldn't take as long. Okay, we're going to go talk about the therapeutic potential, and really it's exciting because I hope I've conveyed to you now in that first lecture. Work in progress, obviously. I mean, it's very complicated, these metabolic pathways, but can you get drugs now to target metabolic reprogramming? Mainly in macrophages are the main cell type I'm interested in, of course. And really see if we can get new therapies and break the efficacy ceiling. Now, my company Citrix, I'm co-founded with 4 others who are real pioneers in the area. We're going after various targets. PKM2 is one big one, for example. And really, to get to the therapeutic bit, I want to re-emphasize a couple of things. The mitochondria become a very interesting sort of place to go with this. Now, PKM2 is interesting, as I mentioned, and there could well be ways of targeting PKM2. But the mitochondria and Krebs cycle became more and more interesting. Lots of really interesting discoveries there. And again, as I mentioned already, the IL-4 activated macrophage has OXPHOS, normal respiration. There it is, going round and round. LPS, you get a very strange change. You get a superglycolysis. But secondly, you get breaks in Krebs cycle. And my lab was the first to show this. Succinate accumulates. And the cycle is broken, and you get HIF. Succinate can drive HIF and drive IL-1. Citrate also accumulates, and that will accumulate. That will cause fatty acid synthesis from citrate, and I think taconate, which I'll come back to, and nitric oxide. So we're actively breaking Krebs cycle here to generate metabolites with downstream consequences. Who would have thought that you'd break Krebs cycle to make succinate to drive IL-1, a really important cytokine, or that you'd break after citrate to drive fatty acids, and as I said, they're inflammatory. They will make prostaglandins. They can tie into nitric oxide, a very important inflammatory. Whereas the IL-4 macrophage doesn't do that. The second thing is, therapeutically, that IL-4-driven macrophage is actually anti-inflammatory and can help resolve inflammation. So that's a good state to be in, to have OXPHOS burning away. And the mitochondria really go weird in macrophages. And this is part of the Warburg. They break up. You get a thing called fission. So the resting macrophage, these are long, smooth mitochondria. They're aspiring. Put on LPS, and look, they broke up into little blebby things. And these are very dysfunctional mitochondria, and that's why it's a big focus. They're pathologic, whereas the ones that are smooth are called homeostatic, which means they maintain the balance. Now, succinate then becomes interesting, and what was the mechanism for its effects? What we found in the end was it could stabilize HIF, and one way it did this is shown here. We made an amazing discovery, if I say so myself, where LPS on a macrophage, the electron transport chain, ran backwards. So in other words, there's a break in Krebs, but secondly, you get reverse electron transport, or RET, R-E-T, through complex 1. And that generated ROS, and the reactive oxygen was driving HIF. And you needed oxidation of succinate by succinate dehydrogenase, complex 2 in the mitochondria. And then the electrons went from succinate through SDH, through complex 1, through RET to make ROS to drive HIF. And a big increase in membrane potential because you can't pump protons anymore, and electrons. So that increase in membrane potential was a feature as well. And that made us think if we block this, and a drug called dimethylmalonate can block succinate oxidation by targeting SDH, and that was potently anti-inflammatory. So we gave mice DMM to block the succinate oxidation, gave them LPS, they made loads of IL-1, DMM in the mice in vivo could block IL-1, and even more excitingly could boost IL-10. So those mice began to make loads and loads of IL-10, and IL-10 is anti-inflammatory. So who would have thought SDH would be a pivot then for IL-1 versus IL-10? And when you have that reverse electron transport, you get loads of pro-inflammatory factors, less of the anti-inflammatory ones. You block SDH, and now you switch and you make anti-inflammatory products, and you might drive homeostasis. Now again, let me just tell you how exciting this is. An SDH inhibitor, and you get anti-inflammatory cytokines. Wouldn't that be marvelous in an inflammatory disease? Because you pivot away from inflammation and towards an anti-inflammatory effect. So the homeostatic macrophage then is low succinate, ATP is coming off the mitochondria, IL-10 is being made. When you inflame that macrophage, succinate now builds up, you get SDH, reactive oxygen, driving IL-1, and this somehow suppresses IL-10. So the mitochondria have been repurposed to drive IL-1 production. And then we wondered were any Krebs metabolites anti-inflammatory. So succinate is pro-inflammatory. And, you know, there's a good name, succinate. And we turn to itaconate. This is a bit like the IL-10 of the immunometabolic world. What is it? It comes off Krebs cycle. So aconitate is ripped away into itaconate, and IRG1 is the enzyme that does it. Now, we found this was really, really interesting because we wondered, would that be a natural form of dimethyl malonate? Could it block IL-1 and boost IL-10? We'd seen a huge increase in itaconate in LPS-activated macrophages. Other labs have seen this as well. Look at that. This is measuring different metabolites, and itaconate goes up hugely, 6 logs higher. What does it do? It's anti-inflammatory. It'll block IL-1 with the data for you there. There's LPS driving IL-1. There's itaconate blocking IL-1. This is interesting. If you knock down the gene that makes itaconate IRG1 in macrophages, you can boost IL-1. So there's the knockdown. So you're losing the IRG1 driving the itaconate, which is great. We gave mice itaconate, and look at this. We protected them from sepsis. Here's mice dying of LPS sepsis. Gave them itaconate, they died. Now remember, this is coming off Krebs cycle. It's a mediator coming off Krebs cycle. Bottom line, what it does is it modifies target proteins. It reacts with cysteines, and there's various target proteins that itaconate modifies. Don't worry about the detail. All of these are pro-inflammatory. Itaconate modifies them and inactivates them. So that explains why we're protecting mice from LPS-induced sepsis. And then lots more work went into itaconate. We showed it could drive NRF2, which is an important anti-inflammatory transcription factor. Another lab showed ATF3 to block cytokines and have an anti-inflammatory here. And there, I can block SDH. I mean, that was also shown. It was like dimethyl malonate, a natural form, to block reverse electron transport. And then most recently, our new data. This is interesting. So the level of itaconate inversely correlates with disease severity in rheumatoid arthritis. This here's the disease score. The higher the score, the lower the itaconate. As the disease score goes down because of therapy, itaconate goes up. Inverse correlation. And look at this. With COVID, there was inverse correlation. So itaconate was reduced with severity. Now remember, it's anti-inflammatory. So that might mean less itaconate means more inflammation in rheumatoid arthritis and maybe in COVID. That provoked us to look at its role in COVID. But two other things I want to tell you about. We also found that itaconate could block NLRP3. Now, as I've been saying for a long time over these lectures, NLRP3 is a key inflammatory pathway. It drives IL-1, drives IL-18, drives cell death. We tried itaconate against NLRP3. This is a derivative called 4-octyl itaconate that gets into cells, that penetrates the membrane. There's the NLRP3 activation. You get lots of IL-1. Itaconate blocks 2 different stimuli. It blocks IL-18. Look at that. There's the LPS-treated sample blocked with itaconate. Very nice. We took IRG1 knockouts that can't make itaconate, put LPS on them, and we got a boost in IL-1. That was more evidence then. See that boost in the knockout versus the wild type? That was great. We took samples from patients with CAPS, this auto-inflammatory disease where NLRP3 is mutated, and again, blocked IL-1 in patient samples. That was great. And then what's the mechanism? Again, it's modifying a protein. It's modifying NLRP3 itself on cysteine. And as I said earlier, going back to this earlier slide, all these proteins get modified by itaconate on cysteine, and that suppresses inflammation. And now NLRP3 is being modified. And that's suppressing inflammation. That's the mass spec of that profile. We got the cover of cell metabolism with our paper about 4 months ago. So itaconate targets NLRP3, a very important anti-inflammatory metabolite. Now, the other thing I want to tell you about then is our favorite disease, if you want to put it that way, COVID-19. Now, how we got onto this was as follows. There was good evidence of NLRP3 in COVID. Lots of macrophages in the lungs, NLRP3 sensing the virus, driving IL-1 and IL-18 and pyroptosis, lots of cytokines being driven downstream, and all the symptoms of COVID began to emerge. And there was good evidence for NLRP3 in this disease. And this is a paper in JXMed, just came out actually about a month ago. Here's NLRP3. You can measure it in various ways. That this big green blob is active NLRP3. This is a macro PBMCs from a COVID patient. Lots of NLRP3, lung cells, lots of active NLRP3 in those. We measured IL-18 ourselves in samples from St. James's Hospital from severe COVID patients. And IL-18 was elevated in the patients compared to control. Now remember, iodine is coming off NLRP3. So more evidence for NLRP3 in COVID was the idea. And as I mentioned, there was an inverse correlation between itaconate and disease severity. Now, all of that tells you that this could be interesting to look at, and especially when this paper came out. Now, this is back in September. And as I mentioned, LPS will drive succinate and reverse electron transport, driving ROS, driving HIF, driving cytokines. A lovely paper repeated our work and used SARS-CoV-2 instead of LPS, and that caused the same metabolic change in infected cells, driving cytokines, driving damage. All of this then adds up to a very simple question: could itaconate be useful in COVID? And there had been a paper on itaconate killing Zika virus. That was from the paper on itaconate. There's Zika. So we wondered, what about SARS-CoV-2? And then remember, I've told you itaconate is anti-inflammatory already. Going back to that slide there, just to re-emphasize that for you. So itaconate will target all these proteins and modify inflammation by targeting them. Secondly, it's targeting NLRP3, the daddy of all the inflammatory proteins. Thirdly, NLRP3 is in COVID. Fourthly, IL-18, a marker for NLRP3, is up in severe COVID. And finally, itaconate's down in COVID. And SARS-CoV-2 causes a metabolic shift that's similar to LPS, driving inflammation. And we've seen LPS drive itaconate. So we put all that together and we wondered, would it work against COVID? And dexamethasone, a well-known anti-inflammatory drug, has been shown to work in COVID. So we kind of combined 2 or 3 things here and tested it. And here's some of the fresh data, especially for you, wonderful people. This is a collaboration with Martijn van Hemert in Leiden. You can infect human lung epithelial cells with SARS-CoV-2 and it kills them. The virus is very pathologic. We pre-treat it with itaconate, and look at this, to stop the cells dying. So it's protecting cells from death with SARS-CoV-2. We measured gene expression from the virus. RDRP is the polymerase. That was suppressed in supernatants from the cells. Another gene called E was suppressed in the lysate from cells suppression. So it's blocking gene expression from COVID-2 itaconate. Okay. And then we got scooped. A paper just came out on itaconate working on SARS-CoV-2. So that was a bit of a shame, but still consistent with our data. Finally, and this is a work in progress, we're just beginning this in some ways. This is a hamster model of COVID-2 infection, hamster model of COVID-19. We infected hamsters with COVID-2. Our collaborator in Leuven did this for us. And that hamsters got really sick. Their lungs were badly damaged. This is a scoring system for lung injury in response to COVID. You get pneumonia, you get edema, you get bronchiolitis, and you get a score. And the higher the score, the worse the damage. There's the uninfected hamsters. Here's the infected ones. These are different hamsters, all showing high scores in terms of lung pathology. We gave them itaconate, and we could protect those lungs. Okay, this is on only one experiment. So, but still, we're trying to do more and more on this now. So what this means is itaconate has potential as a therapeutic. And let me give it to you again. Now, if I was able to write this on the board, I would. What I've told you is as follows. Have a listen and write this down. An inflammatory macrophage, that means LPS, and now it means SARS-CoV-2, 2 inflammatory drivers, okay? Succinate will accumulate. The succinate will undergo oxidation through SDH. You get reverse electron transport through complex 1. That drives reactive oxygen, that drives HIF, that drives IL-1 and other downstream inflammatory targets. So there's immunometabolism going great, isn't it? You're ripping succinate away from Krebs and it's pro-inflammatory, right? Now, secondly, itaconate can build up as well, and that can feed back and suppress inflammation. And it does it in a number of ways. It blocks SDH to stop succinate oxidation. But secondly, it modifies cysteines on target proteins. All these proteins are inflammatory. One of them is LDH in glycolysis, for example. Another one is NLRP3, and itaconate can then be anti-inflammatory. We also think separately it can be antiviral in some way. It can target the virus. We're not quite sure how it does that with SARS-CoV-2. We're working on that very hard. But secondly, it will suppress inflammation in a hamster model of COVID-19 and protect the lungs. And it's doing it probably by blocking NLRP3 and the other targets that we have and blocking succinate. So this effect here in hamsters is explained by itaconate modifying several downstream targets to suppress inflammation. Okay, and that's a very exciting result, we feel. Now, finishing up, I want to say a few more quick things. Why work on immunometabolism? It's scientifically challenging and fascinating, let's face it, but new therapies might emerge. All kinds of prospects for new therapies. Now, why would I say that? Well, there are 2 drugs already used to treat inflammatory diseases. Methotrexate, that's a metabolic reprogrammer. Metformin is a famous drug that's used for type 2 diabetes. That's also a metabolic reprogrammer, and both of these are very anti-inflammatory. Now, metformin blocks complex 1, so maybe it works by stopping succinate. Boosts IL-10 and other anti-inflammatory cytokines. So it's a really potent anti-inflammatory drug, and it's working through metabolism. Methotrexate, so many papers, mechanism unclear. It probably affects carbon-1 metabolism and changes the epigenome, going back to my previous concept. So again, 2 very interesting drugs already in humans. And that tells us we can target metabolism for therapeutic gain is the idea. And that's the bottom line, really. It might be possible to exploit the plasticity of pathways, if you like, for therapeutic gain. You would have thought it might be very toxic and, you might cause damage by targeting metabolic pathways. That's not true. There's really specific metabolic changes happening that might be amenable to targeting with drugs. And we never know, we might get brand new therapies out of this, which would be tremendous. One last example I want to give you is this. There's a drug called dimethyl fumarate, DMF. Do you see it there? Widely used to treat MS, and nobody knows why. It's been shown recently it also modifies proteins in a very similar way to itaconate. So this drug, DMF, could be leveraging a natural process that itaconate regulates by targeting inflammatory proteins and having an anti-inflammatory effect. And secondly, interferon-beta is used to treat MS. That's a great inducer of itaconate. So we're now wondering, is that good for MS because of itaconate? Okay. These are new ideas. Just write them down, okay? And think about them. Use your brains. You're now in the final year of your degree, and I want to challenge you, okay? And I know it's complicated. Any questions, come back to me. But this idea of using drugs to target these pathways then isn't as wild as it seems. So let me now just summarize, because I know it's been a bit complicated. And I might go over some of this again, because I've gone through it quite quickly. Hang on a minute. Taconite stuff. Hold on a second. Yeah, we're really talking about, the succinate business and itaconate again. I want to re-emphasize that, okay? And it is all about mitochondrial damage in the end, right? I'm going back too far. And where the hell is it? Hang on. That's the one I want. So it is all about metabolic reprogramming. And remember, what you're looking at on the screen now is the intense complexity of what happens in a macrophage when it's inflammatory, metabolically, in relation to Krebs cycle. Okay? Not glycolysis. We did glycolysis before. And look at how complex it is. And really, it's a staggeringly exciting business. So let's do it again. Succinate goes up in the inflammatory macrophage. It can leave the mitochondria. It can drive HIF. And then HIF goes to the nucleus and drives IL-1 and other inflammatory genes. There are also succinate receptors on pro-inflammatory cells that activate them. There are things called tuft cells for type 2 immunity. Again, succinate drives those. So succinate becomes a fascinating thing. One thing it can do, though, is it can block SDH. And that stops this reverse electron transport and lowers ROS and HIF this way. So it's a very interesting thing that hang on. That's itaconate. Scratch all that. The succinate can also go through RET and drive ROS. Now, itaconate, that builds up, and it's like a negative feedback effect. So what's happening here is Krebs, succinate, pro-inflammatory. Later in the time course, itaconate goes up, and that's anti-inflammatory and begins to suppress things. And it does it in a number of ways. It blocks SDH to stop that reverse electron transport, to stop the ROS coming out. Secondly, it can also leave the mitochondria and can activate NRF2, which is an anti-inflammatory transcription factor, and multiple other proteins. You can list here NLRP3, very inflammatory. You can list here LDH in glycolysis. Again, glycolysis is inflammatory. So suddenly these 2 metabolites, itaconate and succinate, are the yin and yang in many ways of what's going on. The other thing itaconate does is it drives ATF3, which suppresses IKB zeta, and that will block inflammatory gene expression. IL-6 is over here. So again, isn't it incredible? Intense complexity, metabolic reprogramming, many unanswered questions. We know IRG1 is the enzyme that makes itaconate. We know it gets induced by LPS. We're not quite sure how. I've got a project in my lab. Christian is doing that project. we still don't fully know the full extent of itacanate's effects on these pathways. We've got recent stuff on itacanate affecting coagulation in COVID, for instance. So very much a work in progress trying to figure out what's going on in these pathways. And the connection to NLRP3 then becomes all the more vivid, I think you would agree. And I think the therapeutic prospects, as I said at the very start then, are extensive, and let's get more and more information. This data here is important because this is in humans, and the inverse correlation with diseases, you know, so the less severe the disease, the more itacanate. Again, getting back to my comment in the last lecture, correlation is not causation. CRP goes up with disease severity, see that? And again, then when I saw the COVID results where there was an inverse correlation when itacanates reduced with COVID severity, that was great. And then the fact that the metabolism in SARS-CoV-2, where's that slide? This one, you know, the SARS-CoV-2 is causing succinate and HIF and all these genes. It looks as if any inflammatory macrophage will have these changes. This paper, by the way, said that high glucose is a risk factor for COVID because of this. So type 2 diabetics have very high glucose. And it's feeding this succinate production and RET, you know, and the damaged mitochondria. So even though Krebs is broken, glycolysis still feeds into Krebs and ends up as an accumulation of succinate is the idea here, you see. And that crosstalk then becomes all the more important. So again, I'll just finish and say, you know, all kinds of prospects. These two drugs, metformin and methotrexate, give us all kinds of prospects. And it might be possible to exploit these pathways for therapeutic gain is the bottom line. So, to summarize, in this rapid fire lecture, I spoke twice as fast as I should. I hope you could follow it all. Bottom line is this. Metabolic change is happening inside immune cells. That metabolic change can give rise to very specific metabolites, which then link directly into immune activities. It could be cytokines is the main thing we've spoken about. It could be other things. It's highly complicated, but it's very druggable. You can limit these pathways and see a therapeutic effect. I told you about PKM2. Make sure you get that. That's the key driver of IL-1 when you think about glycolysis. That was the big story there. Secondly, I just told you about succinate and itacanate. They're the three things I want you to know about. And then more broadly about metabolic reprogramming, the Warburg effect, you know, the role of, you know, mitochondrial damage versus normal mitochondria in different cell populations, all that kind of thing, that early stuff as well. And then that will do me, and then you'll get a question that will be along those lines. I'm going to send you a couple of things to read on the reading list, and you'll get these slides as well. So again, thanks for listening, and any questions, don't hesitate to give me an email. Don't be frightened of this area. It's very, very important and exciting. Actually, one last thing. I know there was a big lag there. There was something else I wanted to tell you. There's something that I forgot to put in there. Oh yeah, this one. Just one last little section I left out actually, which I should have put in. Mitochondria, I've been banging on about them at length, haven't I, and saying how important they are, and their change driving various responses. This will be on the reading list. They are the powerhouse of immunity, and I want to tell you one last couple of more things about them. And I kind of mentioned this earlier, but it's worth emphasizing. the mitochondria are generating these signals. That's the bottom line. And it's a very interesting idea that the mitochondria now are responding to inflammatory stimuli, and it could be things like LPS or whatever. Cytochrome C, of course, was the daddy of all that, because that comes out of mitochondria and drives apoptosis. As I mentioned before, the mitochondrial DNA, it's sensed by C gas, and that drives these IRFs, and that induces interferons and all kinds of downstream genes. It can also be sensed by TR9, NF-kappa B, IRFs, immune gene expression, and now the NLRP3 bit, that senses ROS from mitochondria. We think it could be coming off the succinate pathway, and then that drives IL-1, IL-8 and apoptosis. And interestingly, just for completeness, viral RNA sensed by RIG-I, MABs is in the mitochondria, and that can connect to NLRP3 and also drive downstream signals. So there's a little snapshot. Learn that one, because that tells us how metabolic reprogramming is so important for these very interesting downstream consequences. And pyroptosis is a key type of inflammatory cell death. Here's a way to illustrate. Look at that. The cell explodes and releases all its stuff. It's horrendous. Much more out of control than apoptosis would be. So that's something to remember. That side there is important. I'm going to show you a couple of more quick things, actually. This is succinate and RET and ROS. Recent review. We did find reviews. I'm going to put this in the reading list, so I'm going to show it to you. Here's reverse electron transport again, generating mitochondrial ROS. Loads of papers on itaconate in the immune system. Effects on pathogens, really important. That's the antiviral thing. I've shown you these already in the previous section. One last thing. Hang on. It's great to flick around like this. Here's an interesting one. Itaconate can modify JAKs and STATs. You know, and I mentioned those in the cytokine lectures. But I won't annoy you with that because that'll drive you nuts. Yeah, there it is anyway. Look, there's JAK1. We think itaconate can modify JAK1, isn't that cool? Which is interesting in its own right. There's the COVID stuff. We did that already. Yeah, this one. I'll show you this. Very good. Okay, this is the one. So again, the overall concept then from that last lecture with the therapies is to do with cysteine modification and modification of proteins. I told you how itaconate can modify proteins that are inflammatory. Dimethyl fumarate can as well. Fumarate itself can from Krebs cycle. That was a recent thing. Things like GAPDH in glycolysis, gasdermin in pyroptosis. And then these drugs, DMF8, disulfiram is a drug that's used to treat alcoholism actually. That modifies inflammatory proteins. NLRP3, JAK1, we think the 4OI. Now that is mimicking itaconate. That's the itaconate derivative. And that might decrease inflammation and provoke resolution. So we think modification of proteins by metabolites on cysteine could be a very important area to consider as well. There are drugs out there already that can do it, BTK inhibitors and so on. So that was the last thing I wanted to mention. I'll plug those slides back into the main presentation because and you'll have them. And that's it. I'll definitely finish. Now, thank you very much.